Okay, well, we'll move on to our next speaker today for a PhD exit seminar from Ananthan, who will be presenting on investigating the direct and indirect associations between menopause and brain health. Thank you, Ananthan.
Not a problem. Thank you very much for that. Let me just throw this into full screen. Wonderful. So thank you all for your attention and your attendance for this talk, which, as Sonia was saying, is investigating the direct and indirect associations between menopause and brain health. So this is my PhD exit seminar, and what I thought I'd do was I thought I'd start off this talk with a little bit of a metaphor or a visual representation of what my PhD has been. So the PhD has definitely been a journey for me. Over the years, a lot has changed. I think I'm definitely a little bit older. I have more gray hairs, and I'm a bit less well groomed, I think. but overall, I think, you know, on a serious note, like I'm really grateful to have learned so many skills over the course of my PhD. Skills in statistics, neuroimaging analysis, and coding in R. Both of these photos were in fact taken in relation to media stories about my research. So I've been able to develop the skills of science communication as well throughout the PhD. And all of these opportunities have sort of been present throughout my PhD journey. And I've been incredibly lucky to have a supportive team of supervisors and just a team in general that have helped me through it. So I guess what this talk is going to sort of be a reflection of this PhD journey. And hopefully by the end of it, you'll sort of get a sense or a grasp of what the main findings were from the studies from my PhD. It also wouldn't be a journey without looking ahead. So I'll also discuss some steps forward and future directions at the end. So just for a bit of background, my research focuses on menopause, which is defined as at least 12 consecutive months of amenorrhea, which is also known as the absence of menstruation. So the plot on the left shows the distribution of ages that women experience menopause. This is women from the UK. And as you can see, sort of the average age of menopause lies around 46 to 52 years of age. Given that the average age or life expectancy of women in developed countries lies around 81 years, women will on average spend almost 40% of their lives in a postmenopausal state. The figure on the right sort of highlights that an important health challenge that faces postmenopausal women involves maintaining a healthy brain, given that in 2018, dementia was the leading cause of death for women. This graph is from women living in Australia, but also women in the UK. In part, this is due to increased life expectancy in women. However, more research is required to determine what role, if any, menopause may have in contributing to brain health. As noted by the title of this talk, the broad aim of this thesis has been to investigate possible direct and indirect associations between menopause and brain health. But an important question embedded throughout this thesis has been to sort of disentangle how much of any observed effects were attributable to aging compared with the possible effect of menopause. This has been a significant challenge in this area of research, given that both aging and menopause co-occur. As a result, I'll regularly be revisiting this question when discussing my research methodology, statistical analyses, and results. But to better understand the health outcomes associated with menopause, we need to determine precisely what physiological changes occur around menopause. This leads us to my first study, which looks at fat mass changes in women as they progress through menopause. And this was a systematic review with meta-analyses. Notably, for this research question, there was a divide in the literature, with some researchers suggesting that any observed differences in fat mass around this time in quantity or distribution was primarily due to aging, with menopausal status having little to no effect, whereas others reported the complete opposite. The contradictory findings could be due to a number of factors, including the aforementioned sort of like intertwined relationship between menopause and aging. But another possibility is the heterogeneity of measures used between studies when investigating fat mass changes in quantity and distribution. You see, there are many different ways people can measure fat mass. Often body mass index is used. However, whilst this has some benefits, including the ease of measurements and reported accuracy with self-report and measured recall, there are limitations because it indexes changes in body mass, as the name suggests, which also includes lean muscle and bone. So therefore, any changes that we see in fat mass may be confounded by changes in these other components if we use BMI as this measure of fat mass. Other measures include waist circumference, which has been shown to be a useful proxy measure for visceral fat, which is fat around the stomach and organs in particular that accumulates over time. Now, I remember having a discussion with my supervisor about this when we were talking about, studying fat mass more broadly. And I was very confused why people would use body mass index over waist circumference, particularly when focusing on central fat. And he asked me three simple questions, which were, the first one was, what was my waist circumference? At the time, I didn't really know what that was. Then he asked me, what's your weight and what's your height? I guess from these sort of questions, you can sort of see why it is that BMI is so commonly used as a measure, because it's relatively easy. A lot of people know what their weight and height is, and research has shown that there is a high correlation between self-report and measured BMI. However, the gold standard for measuring fat mass includes something called DEXA, which is dual energy X-ray absorptiometry. And this precisely measures total body fat or even regional fat, but also other compartments. You can measure bone mineral density and also lean muscle mass, but I'm just going to focus on fat mass here. So therefore, for this study, we looked at many different types of measures of fat mass to sort of resolve these differences, including body mass index, waist circumference, hip circumference, body weight, total body fat percentage, trunk fat percentage, waist to hip ratio, total leg fat percentage, abdominal and suprailiac skin fold thickness, abdominal subcutaneous fat, and visceral fat. So lots and lots of measures of fat mass were included for this question of interest. So we reviewed both cross-sectional and longitudinal studies that had reported changes or differences in fat mass between premenopausal and postmenopausal women. And studies published up until May of 2018 were included. For the meta-analysis, 201 cross-sectional studies were included, which provided a combined sample size of over 1 million women. And 11 longitudinal studies were also included, which provided a combined sample size of almost 2,500 women. So the main findings from this systematic review with meta-analysis were that there was an increase in fat mass between pre and postmenopausal women across most measures, with the exception of leg fat percentage, which decreased. For example, the figure here on the left is a forest plot, which shows the difference in trunk fat percentage between pre and postmenopausal women. And we can see that this difference was about 5.5%, which was almost a 0.5% increase in trunk fat percentage per year. So as noted earlier, the relationship between menopause and aging, it's quite difficult to disentangle since both progress concurrently. So one approach that we had to address this question included conducting a meta-regression with age as a covariate. And indeed, aging significantly accounted for the unexplained variance. We also compared longitudinal trajectories of changes in fat mass in women aged 18 to 45 with trajectories reported in women in our study. If there was a detectable effect of menopause above that of aging, then we'd expect that the rate of change would vary around menopause. In fact, the longitudinal trajectories reported in our study closely and in some cases exactly matched typical trends for fat mass increases in women aged 18 to 45. Finally, we also assessed whether there was an interactive effect between age and menopause. Essentially what this means is we wanted to see whether there was a difference between the rate of change in fat mass between premenopausal women and postmenopausal women. We found that there was no difference between these slopes, so essentially no interactive effect between age and menopausal status. From these analyses, we therefore concluded that the change in total fat mass was predominantly attributable to age, with menopause having no detectable additional influence. We did, however, observe that there was a change in fat distribution, with increasing central fat and decreases in leg fat. These changes were likely to, at least in part, be due to hormonal shifts that occurred during midlife, when women have a higher testosterone to estrogen ratio after menopause, which previous research has shown has been linked to enhanced central fat deposition. Furthermore, we conducted subgroup analyses, whereby we included women who used hormone replacement therapy, and then we excluded them to see if there was a difference in results. What we found was that when we included women who used hormone replacement therapy, there was a significant increase in body fat percentage and a significant decrease in trunk fat percentage, which was suggested a possible protective role of hormone replacement therapy in preventing or reducing trunk fat deposition, but not in preventing overall fat mass gain. Another important consideration for interpreting these results includes the underlying differences between women who have previously used hormone replacement therapy or have never used hormone replacement therapy. For example, previous research has shown that women who have used hormone replacement therapy, they tend to be more affluent, educated, leaner, and have a better cardiovascular risk profile compared to those who have never used hormone replacement therapy. However, our findings from subgroup analyses align with previous meta-analyses of 8 randomized control trials, which found that postmenopausal women who used HRT, hormone replacement therapy, had less waist circumference and trunk fat percentage compared to placebo. So from all these results together, we were able to conclude that fat mass was indeed higher in postmenopausal women compared to premenopausal women across most measures, with the exception of leg fat percentage, which decreased, indicative of a potential change in fat mass distribution after menopause. However, the change in fat mass quantity was predominantly attributable to increasing age, with menopause having no significant additional influence. Similar to overweight and obesity, dyslipidemia has been shown to be associated with an increased risk of dementia. This is likely related to the positive association between fat mass and lipids. We also noted that there was a lack of quantitative estimates in previous narrative reviews that had described changes in lipids across menopause, which are typically made available through a systematic review of the literature with meta-analyses. This had not yet been done for serum lipids, possibly because the extant literature on this topic may have been too large to systematically review. Importantly, there was also disagreement in the literature regarding changes in high-density lipoprotein levels. So high-density lipoprotein, or HDL, is commonly referred to as your good cholesterol because it sort of transports fat molecules from cells, including those located within artery walls, back to the liver. So some studies have reported that HDL decreases after menopause onset, whereas others reported no change. Given that lipid profiles are highly related to fat mass, particularly central obesity, we used the data extracted from our initial review to address this question. 66 cross-sectional studies were included in the analyses, which provided a combined sample of a little over 114,000 women. Overall, what we found was that postmenopausal women indeed did have significantly higher levels of triglycerides, total cholesterol, low-density lipoproteins, and also total cholesterol to high-density lipoprotein ratio levels than premenopausal women. Similar to our study on fat mass, changes during menopause, we were interested in examining the contribution of aging to differences in lipid profiles. However, we were limited to cross-sectional analyses, given that there was a lack of longitudinal studies that had reported compatible measures for meta-analyses. Despite this, we conducted subset analyses of studies with a mean age difference of 5 years or less between premenopausal and postmenopausal women. And we found that there was no difference in the direction or significance of effects compared to initial estimates on the entire groups. So the magnitude, however, remained the same for most measures, except for triglycerides, it was slightly smaller. Indeed, for meta-regression analyses, this indicated that the mean age difference between groups accounted for a significant amount of the variance. Importantly, as the figure on the left shows, from the studies included in our analyses, we found no difference in high-density lipoprotein levels between premenopausal and postmenopausal women. Therefore, not all differences in lipid levels around menopause were negative. Ultimately, we could conclude that postmenopausal women had an unfavorable lipid profile compared with premenopausal women, which was partly attributable to the age difference between groups. So now that we've sort of precisely understood the physiological changes that occur in fat mass and cholesterol around menopause, we were interested in assessing the association between these changes and the brain. A brain region of interest is the hippocampus, which is shown in yellow here. This also brain image scan is from my supervisor. I was very surprised to find that it all looks normal, so all looks good. So well done, very healthy. But as a bit of a background for the hippocampus, so the hippocampus is a brain region which is sensitive to changes, particularly in the early stages of dementia. So dementia, particularly Alzheimer's disease pathology, is known to begin decades prior to the presentation of clinical symptoms. So given this, the effect of physiological changes on brain health may be reflected in brain volume, particularly the hippocampus as a marker of brain health. Unfortunately, the data set that we used, which was the UK Biobank, it didn't have data on cholesterol and lipids and brain volume at the same time point. However, this was not the case for fat mass. So for the next study, what we did was we investigated the association between changes in fat mass over time with hippocampal volume in women and men. We thought it was important to account for baseline fat mass when discussing changes in fat mass over time. So as a result, we used standardized criteria to categorize individuals into different groups. So on the left in this diagram here, you can see our measures of fat mass included waist circumference, waist to hip ratio, and BMI. Each of these plots also represents the differences between the groups and the different groups. So those who maintained a normal stable weight across baseline and follow-up assessment were the NS group that we see here. Those who progressed from normal at baseline to being categorized as overweight or obese at follow-up assessment were the NO groups. The OS group are those who were overweight or obese at baseline and follow-up assessment. And the ON group are those who are classified as overweight, obese at baseline assessment, however, at follow-up were normal weight. So that's the ON group. So in this study, a little over 20,000 participants were included, and our regression analysis adjusted for covariates that included sex, length of follow-up, so number of years, age, education, vascular heart problems, diabetes, physical activity, smoking, and alcohol intake. So the usual suspects with a question like this. And the key findings from this study were that those who suffered from chronic overweight or obesity, so that's the OS group here, they had a lower hippocampal volume compared with those who maintained a normal level of fat mass, specifically 1.13% lower hippocampal volumes. Interestingly, however, those who were within a normal range of fat mass at follow-up assessment, yet were previously classified as having overweight or obesity at baseline, also had lower hippocampal volumes than those who maintained a normal weight at baseline and follow-up. So to us, these findings emphasize the importance of maintaining a normal weight for brain health, and also suggested to us that the history of weight matters in terms of brain health. But in the context of menopause, what we found was an increase in waist circumference, even from those who were normal at baseline, was associated with a lower hippocampal volume, which was 0.58% lower hippocampal volume. However, this effect was not detected for BMI, which was possibly due to the links between visceral fat, waist circumference, and inflammatory pathways that are associated with the brain. So the next study that we were interested in conducting investigated the association between measures of menstruation history, including menopausal status and age of menopause with brain volume. We assessed the hippocampus, but we also assessed total brain volume in this study. And we excluded women who had a history of hysterectomy, oophorectomy, or had ever used hormone replacement therapy. This was in part because hormone replacement therapy use has been shown to impact the accuracy of self-reported age of menopause. Furthermore, studies have shown that hormone replacement therapy has differential effects on the brain depending on the timing and duration of use. So an important question in this study was to address age as a factor. So we took multiple approaches. We had a little over 5,000 women included in this study. And the first thing that we did was we controlled for age in regression models, in addition to other relevant covariates. Since the age range of postmenopausal women exceeded that of premenopausal women, as you can see in the diagram on the left here, we repeated the analyses in an age-restricted sample of almost 1,500 women aged 45 to 55. We also conducted sensitivity analysis using propensity matching, which is essentially where we compared women who were matched exactly on age and nearest neighbor matched on relevant covariates. We then assessed the differences between these matched groups. So a premenopausal woman who was 45 years of age was also matched to a postmenopausal woman who was 45 years of age, and vice versa. This was less likely, but the distribution changed as the age increased. Surprisingly, all three analyses, which were done to try to account for aging in different ways, showed that postmenopausal women had larger total brain volume and hippocampal volumes than premenopausal women. Postmenopausal women also experienced a greater reduction in total brain volume than premenopausal women. However, this difference was not seen for the hippocampus. Similarly, every 1 year delay in menopause after 45 was associated with 1/3% smaller total brain volume and hippocampal volume. So often larger brain volume is seen to be an indicator of better cerebral health, but not always. There are cases where inflammation can lead to larger brain volumes. Another possibility that may help explain these results is that for some reason, those who had larger brain volumes were more likely to have menopause earlier. So these results may be a reflection of sampling bias within the UK Biobank. However, given the ways in which we accounted for age, we concluded that there was likely an effect of menopause on brain volume beyond typical aging effects. But there were some questions that remained to be answered. So before I get into this, I did put this photo up as another analogy. So at the start of this year, I started bouldering, mostly because I was scared of heights. So I thought it would be, a useful way to sort of overcome this fear. But the more I started bouldering, that led me to rock climbing. And I noticed that there were a lot of similarities between rock climbers and researchers. Every rock, such as this one, is called a problem that needs to be solved. You spend lots of time and energy and effort working out different strategies and sequence of moves required to solve the problem, which is known as sending a problem. And once the problem's been sent, you move on to the next rock to climb. In research, I find it's much the same. Often answers to question or one question can lead to more questions that require more answers. So this is a way in which I sort of frame my thinking, and I frame the future directions of my research. So I guess some key questions that I'd like to explore. I noted in my last study that postmenopausal women had larger brain volumes than premenopausal women, but it's unclear whether this is reflective of better cerebral health. So I think it's important to see if these differences in brain volume translate to functional differences in cognitive functioning, so in cognitive testing. Furthermore, I'd like to study longitudinal changes in brain volume for women who are premenopausal at baseline and postmenopausal at final follow-up. Unfortunately, my most recent study was limited to cross-sectional neuroimaging analysis, as this is what was available in the UK Biobank dataset at the time. As more data and scans are collected in future waves, this question will be possible to answer in this dataset. Also, I've noted a few times that brain volume is one measure of brain health. So another possibility that I'm personally really interested in is using a sort of metric of brain aging index, which can use different modalities to aggregate a summary measure of brain health. In that way, I'd like to compare differences in this brain aging index between pre and postmenopausal women, which can act as a proxy aggregate measures for lots of these different indices of brain health. But overall, I believe that women's health has historically been understudied. So I'm interested in continuing to develop and answer questions that relate to women's health to facilitate better health outcomes. Before I finish up, I have to say massive thank you. There are many, many people to thank, but definitely the 4 people up here are very, very important. As I said at the start of this talk, the PhD for me has been a journey, and I wouldn't be in the position I am today without the help of incredibly supportive people. These include, from left to right, Nicholas Shaburn, Michael Hornberger, Aaron Walsh, and Hussein Tabatabai Jafari, who've all been incredibly supportive in shaping my thinking, challenging me to think scientifically, and holding me to high standards, which I'm incredibly grateful for. So I definitely stand on the shoulders of giants, so I'm very grateful to be in the company of these people in the last few years. So thank you very much. That's it for my talk. If you have any further questions about my research, feel free to ask me now. Or if there's not any time, or if you want to ask me another time, feel free to email me and I'll be happy to discuss my research further. Thank you very much.
Thank you very much, Ananthan, and congratulations on your PhD. Do we have any questions from the audience, please?
I might jump in first, if I may, Sonia. First, congratulations, Ananthan. That was a great presentation. In fact, so great that I thought that since it's being recorded, you should probably cut it into bits and put it on YouTube. So that's something you might want to do over Christmas. I was very excited to be reminded of some of the results that I had forgotten, and think about the interpretation. I mean, particularly in figure 1, I could see all the signs of wisdom developing, the hair lengthening, graying, glasses growing, and possibly already a slight stoop. All the indices that suggest, the development of a professorship. And so I was generally very excited. I must say, I was a bit disappointed afterwards to learn that it was all attributable to aging, but nevertheless,
after all these years,
well,
I can't believe I still fell for that. I was on the edge of my seat waiting for the question.
On a more serious note, it's important to address questions like this, but it's also, the question is, so what do we do about it? And so, what would be, how do you think, in two ways, as a researcher, what can you do to find ways of changing these trajectories? And as a communicator, how do you think you can communicate these findings to women so that they're, empowered to lead as healthy a life they can, given the limitations of aging processes, and not just women, men as well, but here we're talking about menopause.
Yeah, no, thanks for the question, Nick. It's great point. I guess from my experience in science communication, especially in particular the systematic review and meta-analysis of fat mass changes in women, I had on more than one occasion women coming up to me and saying, well, I didn't have any increases in fat mass around this time period. What do these results really mean for me? And so a challenge in science communication is sort of communicating these scientific concepts to the public. For example, the fact that these findings are based on a population level and may not apply to individuals in particular. However, at a population level, when we look at women's health, these are the trajectories that we see. So in regards to your question of what we can do, I think an important thing is informing practices of, if we know that, whether it's aging or menopause, if we know that there's this risk of an increase in fat mass around midlife, which we know is associated with negative consequences in health, such as dementia or cardiovascular disease, then we need to make sure that particularly clinicians are able to intervene at early enough stages so that dietary or lifestyle factors are implemented to ensure that people are living healthy lives and maybe mitigate some of the negative effects that occur in midlife. So I think that's a key. And secondly, I think that's where science communication is so key in getting these messages out, and so that women who are now more aware of the changes that are occurring around menopause have more knowledge and are empowered as a result. So I think that's the key points.
Thank you, Nick. Is there any other questions from the audience, please?
I have a question. You were saying that later menopause, does that lead to lower or higher brain volume, or can you remind me what that was?
Yeah, so women who had menopause earlier had larger brain volumes than women who had menopause later.
Okay. Are there any factors associated with earlier or later onset of menopause, lifestyle, or is it just completely dominated by genetics? Or
yeah, no, great question, Mitch. I mean, it's there's a lot of factors that are being studied. Unfortunately, it's not very clear cut. So a few of the main leading factors that have been looked into. One is in terms of lifestyle factors. Smoking has been studied heavily. Research has shown that women who smoked, who have a previous history of habitual smoking, often have menopause a year earlier. So in our regression analyses for our models here, we did adjust for this factor of smoking, whether women had smoked before or after. So this would have sort of helped account for that. But there's definitely other genetic factors, and mother's age of menopause can be an influencing factor. So if the mom has had a menopause earlier, this is predictive of the daughter having menopause earlier as well. However, this seems to be the only case. If mothers have menopause in the normal age bracket of 46 to 52 years, this isn't necessarily predictive of the daughter's age of menopause, so early menopause in particular. And so there's lots of reasons for this factor. It could be biases in women who recall menopause. So the fact that people who are aware of when their mum has undergone menopause. There's a lot of research that shows that a significant proportion may not know, and that could be due to other social and cultural factors. So lots and lots of factors that influence it, but those that we could control for, we did in this regression analysis.
Great, thank you very much, Mitch. If there's no final questions, we might end there at 1:30. So thank you. Please help me in thanking our speakers again and wishing them all the best with their PhDs.
Thanks, everyone.